Wilt u gaan staan? U kunt wel plaatsen.
I hereby open this academic ceremony in which Mrs. Lambrichts will defend her academic thesis, Cerebral Microvascular Dysfunction in a rat model of heart failure with preserved ejection fractions. I welcome you and your partners and wish you lots of luck. I also welcome your supervisors, Professor van Oosterbrugge, Dr. Fouquier, of course the members of the opposition, whom you will meet later, and of course your family, colleagues and friends here on site and online. But before we start the opposition, I give you the opportunity to tell your family and friends what you have presented in your thesis.
Thank you. Dear Pro-Rector, dear members of the committee, dear promotion team, dear family, friends and colleagues, thank you all for being here today. In the next 15 minutes, I'll share with you what I have been working on in this past 5 years. The title of my PhD is Cerebral Microvascular Dysfunction in a rat model of heart failure with preserved ejection fraction. So let's start with breaking it down a little. We'll start off with about the microvasculature. This refers to the smallest vessels in an organ. And these are most important because the small vessels are responsible for exchanging oxygen and nutrients to the surrounding tissue. The cerebral microvasculature refers to the smallest vessels in the brain. Now, the brain is a very special organ. While it only contains 2% of the total body weight, it receives 15 up to 20% of the heart's output. And this is distributed to the brain tissue by a total network of 600 kilometers of vessels. Now, the vessels of the brain are a little bit more special. They are supported by supporting cells, and they make sure that the vessels are balanced, but they also regulate blood flow. They make sure that the vessels are able to dilate or constrict depending on the need of the surrounding tissue. In addition, there is a second barrier of star-shaped cells protecting the brain. Together, these cell types form the blood-brain barrier, and it protects the brain from harmful substances. In addition, these cells also express several transporters that only allows a few molecules to enter the brain environment. Now, why does the brain need so many blood vessels? It requires a lot of energy to perform difficult tasks like planning, organizing, but also for making decisions and memories. So what happens if the small vessels in the brain become dysfunctional? This is called cerebral small vessel disease, or in short, CSVD, and it is one of the leading causes of vascular dementia. CSVD is often diagnosed in patients with high blood pressure, obesity, and diabetes. So dysfunction of the small vessels of the brain can have big impact on the brain function. It can lead to a smaller brain because there is damage to neuronal tissue, but there is also a lower blood flow to the brain. So what about the heart? Because these risk factors are not only affecting the brain. They can also lead to heart failure with preserved ejection fraction, or in short, HFpEF. In the heart, it can lead to a bigger and stiffer left ventricle, and therefore it cannot relax properly anymore, but there is also inflammation and a lower blood flow. So it seems that in both CSVD and HFpEF, a lower blood flow is present in both organs. Could the microvasculature play a role in this? It has been suggested that microvascular dysfunction could be an underlying mechanism in both diseases. In addition, cognitive decline has been observed in heart failure patients. So why is it so important to study these two diseases? For both of them, there is no therapy available. Patients can only be helped by treating the associated comorbidities, and diagnosis often occurs late because the symptoms are vague and overlapping with other pathologies. More than 20% of the dementia patients are diagnosed with vascular dementia that is caused by COPD, and more than 50% of all heart failure patients present with HFpEF. That's why we wanted to study the cerebral microvascular dysfunction in a rat model of heart failure with preserved ejection fraction. So how can we study the microvasculature? First of all, we can look at the function of the vessels. If this becomes dysfunctional, vessels will not be able to dilate properly and they cannot fulfill the needs of the surrounding tissue. Secondly, we can look at the number of vessels. Since there is a lower blood flow, it is also possible that the number of vessels decreases in the organ. And lastly, we can look at the integrity of the vessels, or more specifically, the blood-brain barrier. When vessels become damaged, they become leaky and harmful substances can enter the brain and cause further damage. In Chapter 2, I studied the vascular contribution to cognitive impairment in heart failure with preserved ejection fraction with TRPV4 and KLF2 as key mediators in neurovascular dysfunction in the ZSF1 model. Now, some of you may have heard me talking about these famous rats. There are two types. The lean are born with high blood pressure, and the obese also develop high blood pressure, but in addition, they naturally develop obesity, diabetes, and HFpEF. We wanted to see whether the obese rats with HFpEF also develop cognitive impairment. For this, we performed a series of behavioral tasks, and we could conclude that the obese animals became a little bit more anxious towards the final time point compared to the lean animals. Working memory was not affected, but we did see that short-term memory was impaired, as well as the spatial learning and long-term memory. So from these results, we could conclude that indeed the obese animals with heart failure developed cognitive impairment. Alongside the cognitive decline, we also detected neurovascular dysfunction in the obese animals. Now, to understand what this means, we need to go back to cells that constitute the blood-brain barrier. These are not the only important cells in the brain. There are also neurons and resident immune cells, and together these form the neurovascular unit. This is actually the place where neurons talk with the blood vessels. In healthy circumstances, there is a nice balance between the blood supply and the need of the neurons. However, when cells of the neurovascular unit become damaged, this balance is disturbed and the need of the surrounding of the neurons cannot be fulfilled. We wanted to look at the neurovascular unit in the ZSF1 model. For this, I performed surgeries to thin the skull of the animals, and we could then image the blood flow with a special laser. Of course, we also wanted to look at the function. So during imaging of the cerebral blood flow, we would stimulate the whiskers, and as you can see in this video, the blood flow would then increase to this specific region. Now, in the obese animals, the increase in blood flow was much lower compared to the lean animals, and therefore we could conclude that the neurovascular function was disturbed in the obese animals. Of course, we also want to understand what is causing this neurovascular dysfunction. We identified two genes, TRPV4 and KLF2, as important mediators of this process. For this, we isolated the smallest vessels in the outer layer of the brain, and we looked at the gene expression profile. We looked at which genes are lower or higher expressed in the obese animals compared to the lean at the last 2 time points in our study. We then identified, or we found, that TRPV4 and KLF2 were actually much lower expressed only in the obese animals, and this was not detected in the lean. Now, these two genes are normally vasoprotective. They are involved in regulating blood vessel formation, inflammation, but also blood-brain barrier integrity. So what happens if these genes are lower expressed? It can lead to a more pro-inflammatory environment that can cause damage to the brain tissue, but it can also cause damage to cells of the blood-brain barrier. Therefore, vessels can become leaky, and components from inside the vessels will leak into the brain tissue, causing damage. This is what we have also observed in the brains of our ZSF1 rats. We did not detect an increase in the number of leakages in the obese animals, but we did see that the leakages were much larger compared to the lean animals. We have now looked at the function of the vessels and the integrity of the blood-brain barrier, but what about the number of vessels? Since there is a lower blood flow, it would make sense that there is also loss of vessels. We can study the vascular network of the brain and the heart through microscopy. We would usually study thin sections of the organ, and like you can see here, we can stain the blood vessels with a fluorescent dye and then image it. But to understand the full network, this would be very time-consuming, and we can still miss information. That's why in Chapter 4, we wanted to study the 3D visualization and quantification of the cerebral and cardiac microvasculature in a model of heart failure and cognitive impairment. So again, we studied the lean and obese ZSF1 rats. The obese, we now know that they don't only develop heart failure, but also cognitive impairment. And we wanted to look into the microvasculature of the brain and the heart. But before going to this 3D image, we can fluorescently stain the blood vessels before isolating them. And then we place the brain and the heart in some chemicals in the lab for a couple of days, making them completely transparent. The heart and the brain blood vessels can then be imaged with a fancy microscope, of which this video is the result. After analyzing these data, we actually did not find a decrease in the number of vessels, but we found a mild increase in the number of the smallest vessels, which is a bit contradictory. When we look into the literature, there are actually varying reports about this. Some report an increase in the number of vessels, some report a decrease. And this not only depends on the methods, but also on the type of animal model that was studied. The pathologies can be induced through medication or surgeries, but it also depends on the time point that the animals were studied and how far the disease was progressed. So do numbers tell the full story? In Chapter 4, we found a mild increase in the number of the smallest vessels, but we also showed that they are not working properly. So it is possible at a very late advanced stage of the disease, the number of microvessels can indeed decrease. But at the stage before that, it is possible that the density actually increases as a compensatory mechanism for the long-term exposure to comorbidities. So for future studies, it might be much more interesting to actually detect mild changes in the dysfunction of the vessels rather than the irreversible structural damage that occurs at a late stage. This brings me to the take-home message. In this research, we've shown that the obese animals not only develop heart failure with preserved ejection fraction, but also vascular cognitive impairment. This was accompanied with a dysfunctional neurovascular unit, and we identified two genes, TRPV4 and KLF2, as important mediators of microvascular dysfunction in CSVD. In Chapter 4, we highlight the 3D microscopy as an important tool to study the total vascular network. We found a mild increase in the number of microvessels, but we've also shown they're not working very well. So it is possible that there are changes in the programming of the vessels because they're trying to adapt to the long-term exposure of comorbidities. So to conclude, we have shown that microvascular dysfunction is indeed a key driver of disease progression. And with this, I would like to end my presentation and give the word back to the prorector.
Thank you, dear candidate, for a very clear presentation. We are now going to start the opposition, and the first opponent is also the chair of the assessment committee, that's Professor Schroen. And she's Professor in experimental cardiology, diastolic heart failure at the Department of Physiology at the University of Maastricht. Professor Schroen.
Thank you so much, dear candidate. Congratulations on this beautiful thesis. I would like to also congratulate your supervision team, and it was a real pleasure to read, and I also learned a lot, so it was a mutual benefit situation. I would like to start with asking one of your paronyms to read Proposition 1, just to warm up a little bit.
When the heart weakens, the mind begins to fade.
Yes, so that's actually core to your thesis and everything you write. So I thought you must have thought about this. And just a simple question that I have is, what do you think is the chicken and what is the egg?
Highly esteemed opponent, thank you for the compliments and your question. I think this is a question that will continue for a very long time. I think in this model of comorbidities, a lot of things are happening. The heart and the brain are not the only organs being affected. There are also other organs affected. And in our model of the ZSF1 rats, we found that heart failure and cognitive decline are happening at the same time. So at this point, I think it's difficult to point which of the two becomes dysfunctional first. I think it would be really interesting to study it at earlier time points and really focus on the dysfunction of the vessels to see where it starts first.
Yes, I completely agree with you. My own conclusion was that these are simply two eggs from a very unfortunate chicken. Mm-hmm. So about the ZSF1 model, you combine indeed, this model combines prevalent comorbidities. I think it's an excellent model of HFpEF, and as you have shown, of cognitive decline. And I have some questions about the findings that you present. For example, you compare lean and obese rats, but lean rats also have hypotension, as you presented to us. So wouldn't you expect some impact of this hypotension on cognitive functions? Because you compared lean and obese and not took into account the control rats.
Mm-hmm. Yeah, indeed. The lean are also born with hypertension, so it's not a full healthy control to compare to, but this is not possible with the ZSF1 strain. So for the cognitive testing, indeed, we see that at the final time points, especially during the Barnes-Maze task in the spatial learning, the difference between lean and obese becomes a bit smaller. So it is possible that they're also developing some cognitive decline since it is known in hypertension. But the obese ZSF1 rats, they develop it much earlier and much more severe compared to the lean. So to understand which mechanisms are causing this, it is still fine to compare to the lean rats.
Mm-hmm. Yeah, I understand. Because in the obese rats, you already find atrophy in the brains at a very early time point. So how do you explain that?
Yes, we were a bit surprised with this as well, but we think because they're not only born with hypertension, but they also become obese quite early and start developing diabetes from 10 weeks old, that perhaps the combination of this can also slow the development. So perhaps the processes are already going on, and this limits the full development.
Yeah. That's very well possible. So also early life events are quite important.
Mm-hmm.
That's also important to know. I'd like to continue with this model, and I would like to talk about sex differences, because you used male rats, and as you know, there is a clear sex effect of HFpEF, so females are more susceptible. I don't know what about cognitive decline. Is there any sex difference?
Yeah. Initially, we wanted to include the female rats, but when writing the ethical approval, there was no evidence yet that the female ZSF1 rats developed the HFpEF the HFpEF disease in the same way as the male rats did. So that's why we only focused at the male animals at that point. Later on, studies were published with female ZSF1 rats, and indeed they see that they do develop it, but less severe, and also the time at developing differs slightly. Still, they only used female animals that were not ovariectomized. So HFpEF usually develops in women after post-menopause. So I think this is an important factor also to take into account and to perhaps study the ZSF1, the female ZSF1 rats after ovariectomy.
I agree with you, and but I'm curious for your ideas on what. How did I phrase this? What new territories do you expect to uncover with such an approach, like with the ovariectomy?
Sorry, could you repeat the question?
So what would you expect to uncover further when you also ovariectomize the rats?
That's a good question. Well, initially I would think that similar mechanisms would be driving the disease because it's not only hypertension, and I think the estrogen is known to protect females from hypertension. So in the ZSF1 rats, it's also obesity, diabetes, and a combination of this all. So I think in general there is a lot of stress on the vasculature and especially on the endothelial cells. So I would expect to find similar results.
Mm-hmm. Yeah, and hormonal influences are going to be important. Yeah, great. Do I have time for one small question? Yes. So I was also very interested in your societal impact paragraph. And on page 139 you write, and I quote, "As HFpEF and VCI manifest as a multi-system disease, treatment of each patient using a unique pharmacological approach is very unlikely." Can you explain to me what you mean by that?
Thank you. Yeah, indeed. I think the patients in the clinic, there is a lot of variation. Also, when they want to diagnose CSVD, for instance, some already see like a lot of structural damage in the brain, while in some patients it's mostly cognitive declines. So I think also the therapy should be adapted to this, and I think patients are already being treated with hypertensive and diabetic drugs, and it's been shown to have positive effect on the vasculature. But I think a combination of multiple therapies that stabilize the vessels is needed to get this.
Yeah, absolutely. And indeed, these antihypertensive drugs are not sufficient to prevent heart failure from happening, for sure,
No.
also not brain issues from happening, so we need something else to top up. And do you believe that it's going to be like a one-size-fits-all for brain and heart?
Well, since it's a similar mechanism that is probably happening in both the brain and the heart, I think if a combination of treatments that improves the general health of the vasculature, I think it will indeed improve in both brain and heart.
Okay, well, thank you so much, and I'm giving the word back to the pro-rector.
Thank you.
Thank you very much, Professor Schoen, for your opposition. Then we come to the following opponent, who's also a member of the assessment committee. That's Professor van den Hove. He's professor in neuroepigenetics at the Department of Psychiatry and Neuropsychology at the University of Maastricht. And today he has also accepted the role of secretary, for which I'm very grateful.
Thank you very much, and my pleasure. First of all, dear candidate, and also your promotion team, my compliments and congratulations on this very nice thesis, which I read with great pleasure. I also love the cover, I have to say. And I also like your presentation quite a bit, so nice. I have a few methodological questions, particularly on the behavioral testing. So let me maybe start with the novel object recognition test, where you described that during all the tests, the animals were housed socially, except for this test, where you housed them individually. And I was just wondering why you opted for this, and to which extent you think this may affect the outcome.
Highly esteemed opponent, thank you for the compliments and your question. They are indeed socially housed during most of the periods. Why they were housed individually during this test is because it was standard in the protocol, because they tend to perform better during this test when they're housed individually, so that's why we only did it during this time.
So you actually mean, because there's different protocols there, sometimes people, I mean, the housing is actually socially, but then 5 minutes before you start the test, you then take one of the animals out of the cage and put them in an empty cage, also without sawdust or anything, but this is different. You actually, during the whole object recognition test, so because you have the habituation phase, and then so during this whole period they were housed individually.
Yes.
Okay. Because that may also induce stress, of course. What could you see with the animals also when you put them back together after this test, or was this the last test that you performed before you euthanized the animals?
No. After the novel object recognition task, we performed the Barnes maze.
Yeah. And then for that test, you actually housed them together again.
Yes.
So could you see anything in terms of the behavior in the cage? Because as you just explained, you only dealt with male animals.
Yeah.
And when you put them back together, yeah, they like to show who's the boss. Did you see anything like this in your cage, that there was fighting or things like these?
No. Luckily, the ZSF1 rats are very sweet animals, and we did not observe any aggressive behavior towards each other. So they were housed with 2 to 3 animals per cage, and we didn't see anything strange.
Okay, that's good to hear. All right. I still think that also the isolated housing may have induced some stress. Nevertheless, that's the same than for all the groups you of course included. A few other questions on the same test. One of the things was you use certain cutoffs for the exploration time. It's very common. You want the animals to explore the objects properly, otherwise they cannot remember them. So that makes absolute sense. But then you use 5 seconds for the first trial, and then you have this 1 hour interval, and then you do the second trial, and then you use 8 seconds as the cutoff. Were these values arbitrary, or did you base it on literature, or why did you take the 5 and the 8 seconds? Very short.
This was also because it was a very optimized and very much used method in the neuroscience department, so that's why we took it over.
I think it's also well defendable, and I have a slightly bigger problem with the next one, or at least I'm very curious. So you actually, to measure their behavior, to measure the memory, you let them explore the new object, or the old and the new object in the second trial for a while, but you decided to then only base your memory score on the first minute of that trial. And could you explain why you only used the first minute and not, let's say, the complete trial duration?
Yeah, we first started with analyzing the full 3 minutes, but then we saw that the variation was even higher, especially the lean, because they were sometimes escaping from the arena. So in the first minute, they were focused the most, and the results gave less variation in the data, and we still saw a clear difference between lean and obese. So that's why we went for the 1 minute.
All right. Yeah. Thank you very much. Very clear. And that bridges actually to the other behavioral test, which is the Y maze. Because there I would have chosen a shorter time period to look at the data. Because can you maybe briefly explain what the Y maze does?
Yeah, the Y maze looks at the working memory of the animals, and here the animals are placed in a Y-mazed arena, and we place them on one of the arms, and then they need to alternate in the 3 arms to each time explore the new arm.
Yeah, so if they are on arm number 1, let's call it like that, they go to number 2, and then they come back to the middle and think, okay, I've already seen 1, let's go to 3. Yeah, that makes sense, and that's something you test. So every time they come back to the middle, you compare what they do to chance level, which is 50%. Now there, I would think, hey, I've seen arm number 1, and now visited 2, I go to 3. And then the next time they come back to the middle, I just visited 2, so let's go to 1 again. But after 5 or 6 times, I don't think they care anymore. And that's not working memory, to my opinion, anymore. If you then have seen every arm already a couple of times, nothing is new anymore, if you get my point. Huh? And actually, you do see a difference in the number of arm entries between conditions, do you? So I was wondering whether you could look at those data again in a different way to really look at working memory, because actually also in your lean animals at the last age, you don't find intact working memory according to how you analyze it, correct? Is it something you expect because it's also a model of hypertension, or is it something you didn't want to see?
I think indeed 6 minutes is quite a long time, but we still use the full 6 minutes because the number of arm entries was not super high, and it only decreased towards the end. So we could not limit the amount of time, because therefore otherwise we had to even exclude more animals. I think at the last time point, indeed, we see an impaired working memory in the lean animals. And I think since they are hypertensive, it is possible that it could be impaired at this late stage of the study. Normally, we would exclude animals with an arm entry lower than 10, but we reduced this number because the arm entry was so low in the obese animals. Yeah. So I think this was not the right test to assess the working memory for these
I fully understand.
Yeah.
My time is up, so that's why I'm interrupting you, sorry. Actually, I think the paper is still under review. I would actually not correct for time or anything like that. I would look at the first couple of arm entries. So that's independent of time and exploration activity. So you could actually, per chance for a triad, so per chance for making the right call, you can actually plot your data. So you can look, for example, at the first 10 occasions. And that doesn't matter whether for one animal that takes 3 minutes and for the other animals it takes 1 minute. But you actually look then at an equal number of opportunities for them to make that 50% call, if you get my point.
Yeah, I see.
So, but anyway, I'm very happy with your answers, and I give the word back to the program.
Thank you.
Thank you very much, Professor van den Hove, for your opposition. We now go to the following opponent, who's also a member of your assessment committee. That's Professor van Zandvoort. And his chair is Advanced Optical Microscopy in the Department of Genetics and Molecular Cell Biology in the University of Maastricht. And he's also working at the Uniclinic in Aachen, and that is in Germany. Professor van Zandvoort.
Dear candidate, first of all, congratulations with your thesis, and also congratulations to your promotion team. You've written a very thorough thesis with a beautiful introduction, I must say. But you understand, as a microscopist, I have to ask questions about that part of the thesis and that part of the introduction. As you know, I'm focused on multiphoton microscopy, two-photon microscopy. And my questions are therefore from both chapter 1 and chapter 4, where microscopy is strongly involved. And on page 22, you mention as one of the advantages of multiphoton microscopy, penetration depth. You give a comparison of the various techniques on focal multiphoton and light sheet microscopy. Could you indicate to me more in detail what penetration depth you expect in brain and heart for confocal, two-photon and light sheet microscopy?
Highly esteemed opponents, thank you for your compliments and your question. For the confocal, I think you also mentioned it, we have worked with brain sections from 30 to 50 micrometer, and I think actually 50 micrometers is really the maximum, because otherwise you will have problems with antibody penetration. For the two-photon microscope, we also did a few experiments with this to optimize the tissue clearing, and I think we imaged up to 400 micrometer. And for light sheet microscope, I think the limits they're endless, because they have been clearing whole mouse brains, but also whole animals. So I think they can, with the right clearing method, image very deep into the tissue.
Yeah, and that brings me exactly to my point. It depends on the clearing,
Yes.
how deep you can penetrate. So if you would do on those well-cleared tissues multiphoton microscopy, you would also get deeper. So when you compare the light sheet and the multiphoton, the difference should be in the same type of tissue. And that brings me to this next question. So when you do light sheet microscopy, you also miss some possibilities that you have with multiphoton microscopy. Could you extend on those?
Sorry, could you repeat the question?
Yes, so if you do light sheet microscopy,
Yes.
you can image fast, because that's the main
Yes.
advantage of light sheet microscopy.
Yes.
But you also miss some possibilities that you have with multiphoton microscopy. Think about, for example, penetration depth, but also about intrinsic signals like autofluorescence, third harmonic, second harmonic generation.
Mm-hmm. Well, I think I disagree, because with the two-photon, I think you have a higher chance for bleaching the tissue as well. And I think with light sheet, sorry, I forgot the question.
Whether there are other advantages of multiphoton that you miss with light sheet microscopy?
Well, I don't think so. I think light sheet gives much more information when you image the tissue, actually.
Well, the difference would be the intrinsic signals that you miss with light sheet microscopy. So, for example, the context of in the brain, lipids, etc., that kind of thing.
Okay.
When you say that light sheet would be the method of choice because of bleaching and speed, what if you would image with multiphoton with the same resolution, with the resonance scanning, and with the same Z-stack step size? Then it would also be much faster.
Could be. I'm not an expert in the two-photon microscopy, but I do think that the light sheet has a better axial resolution. So I think, in total, it would give better results.
Yeah, we can discuss that later.
Okay.
But, on page 23, you indicate that you use an injection-based gel staining of the blood vessels. That is possible in animals, but I don't see you do that in humans. So how would you proceed if you would do the similar study in human tissues from patients?
Yeah, then indeed the gel BSA FITC perfusion is not possible. But there are a lot of protocols being optimized currently for human tissue. I think the iDisco Plus, but also the angiomesh from a colleague is really well optimized for clearing human tissues, and they're optimized also for a better antibody penetration into the tissue. And I think that's the only way to really study it in a 3D manner.
Yeah, and smaller antibodies also that are being developed. Okay, so my next question is on chapter 4. That is on page, let me check which page it is. I just know the figure number by heart. Page 107, figure number 4. So you claim there's a statistical relevant difference between the middle group, 22 to 33 weeks, and the other groups. To be honest, I think the differences are extremely small, and especially the difference between lean and obese are very small. So small that I wonder how far they are determined by simple outliers. So for example, in figure G, there is clearly an outlier, and that might be also a cause of the difference that you observe. So what is it based on that you see that large difference?
In figure 4 on page 107. The difference within the same group?
Yeah. So lean versus obese. Let's stay at that. For example, at 22 to 23 weeks. Sorry.
I think at 22, 23 weeks old, the variation is not that high, but indeed at the first time point and the last time point, the variation is higher because the power is less. We had to exclude some field of view because the perfusion was not optimal to analyze. And I think this might have led to a higher variation at these time points.
Okay, I think my time is up when I look at the prorector. Okay, thank you.
Thank you, Professor Zandvoort, for your opposition and your good observation. The next opponent is Professor Bronkaas, also a member of the assessment committee. She's Associate Professor Ischemic Stroke, Stem Cells and Angiogenesis in the Department of Cardio and Organ Systems in the Biomedical Research Institute in Hasselt, in the Hasselt University, and it is in Belgium. And thank you for coming all the way from Hasselt.
Dear promovendus, I want to congratulate you with your nice work, also your promoter. And I have been following you for a long time because you did internship in my lab. And I also am glad to see some old students in the public. But I also am here to ask you some questions. So on page 91, you state that there is a species differences between on the TRP4 channel. So you state that TRPV4 is a major TRP channel regulating the BBB permeability in rodents, but this is not the case for humans, where you have the TRPV2 channel. So now you did all the research in rats, and now you know this. Do you think of another model that you could use to tackle these differences?
Highly esteemed opponent, thank you for the compliments and for your question. Indeed, some papers refer to TRPV4 as also an important channel that is more important in humans. However, I looked further into it, and indeed, in humans, they still report that TRPV4 is the main regulator in mechanotransduction pathways. But I think to be complete, it would still be interesting to have to look at all of them for translational reasons. I think to further study it, it would be interesting to look at it in an in vitro model, to use human endothelial cells, and to look at inhibitors for this.
Okay, thank you very much. I have another question on the RNA-seq data. So you used, you isolated brain vessels. You took the whole brain vessels, but you didn't do any max sorting to specify the endothelial cells. And also related to that, you didn't take into account the endothelial cell heterogeneity, because you have also arterial endothelial cells and venous endothelial cells. So do you think these choices really affect the quality of your data, that it would be different if you changed it?
Indeed, it's not ideal indeed that there are also parasites and astrocytic end feeds in the data that we analyzed. But at that point, a collaborator was optimizing the single nuclei RNA sequencing on brains of the ZSF1 rats, but the protocol was very difficult to optimize, and she could not get enough endothelial cells to perform the experiment. So that's why we isolated the microvessels from our rats. Now, to select on the different types of vessels, we did optimize the protocol to only isolate the smallest vessels, and we also checked this later on with immunostainings. We measured the diameter, and they were around 5 to 6 micrometers, so this means that we're only looking at the capillary microvessels, and I think this still is a nice representation for this.
Yeah, so nowadays people go really crazy with the transcriptomics. You have single cell, you have single nuclei, you have spatial, but you also have bulk RNA-seq. Do you think it's always better to go higher resolution, or do you think bulk RNA-seq is still good these days?
I think for our research, it still gave a nice indication, because we did identify some genes that were only regulated in the obese animals and not in the lean. So we could conclude that the mechanotransduction pathways were highly affected in this model. Of course, it is also always more interesting to look at cell-specific changes. So I think it depends on what you are looking for at that time.
And then my last question, because my time is up. So you see in your model that there is vessel impairment, but on the other hand, there is compensation, you have more vessels. So my question is, and I had to ask an ischemic stroke question, what would happen if at 22 weeks you will induce an ischemic stroke in the normal rats, the lean rats, and the other model, the SVPT rats? What would happen, do you think? Which of them has the largest stroke?
Which has the largest stroke? I'm not sure. I think both animals are susceptible to it. But I think the way they will react to it will be different. In CSVD, we don't see a lot of infiltration in immune cells, and I think with a large stroke, this would be the case. So I think also pathways would be differentially regulated compared to only the regular obese ZSF1 model. I hope that
Could you
answers
have
it.
more BBB leakage in one model? That's also important.
Yes, I think the BBB leakages are anyway largely present in the ZSF1 rat model. So I think by inducing a large stroke, it would only worsen. And stroke is known to be associated with a lot of immune cell infiltration. So I think this will only make things worse.
Okay.
Thank you very much for your position, Professor Bronkaers. Now we come to the following opponent, who's also a member of the assessment committee. That's Dr. Brooks. She's assistant professor in immunology, specialized in neuroimmune interactions in the Neuroimmune Connections and Repair Lab at the University of Hasselt. And thank you also for coming all the way from Hasselt.
Dear candidates, first of all, congratulations on your work. I particularly enjoyed your presentation. It was very clear, so also congratulations to the promoters. Can I first ask one of the paranims to read proposition 2, please?
The presence of comorbidities is associated with cerebral microvascular dysfunction.
Thank you. Indeed, so you studied many different comorbidities, I would say, in your rat model. So which comorbidities exactly, can you sum them up, are present in your obese model?
Highly esteemed opponent, thank you for your compliments and for your question. Indeed, the obese ZSF1 rats develop a lot of diseases. It starts with the high blood pressure, of course, which is very early present in the disease progress. They have a leptin receptor mutation, so therefore they never stop eating and they become obese around 5-6 weeks, I think, and they develop diabetes around 10 weeks. They develop heart failure with preserved ejection fraction around 20 weeks, full heart failure. They also have kidney failure. They are known to develop it around 12 to 16 weeks. And then, of course, the cognitive impairments. But yeah, it's a chronic systemic inflammation model, so I think a lot of organs might be affected as well.
Mm. And so the primary outcome of your studies was to study cognitive function or cognitive decline. So just a question about your study setup. Do you think it's important to be able to tease apart the effects of the different comorbidities, or as you done, to look at this as a whole?
I think when we look at the clinical situation, a lot of patients don't present with only one of the morbidities, but it gradually develops into a whole list of things that are going wrong. And I think the status of one rat model is a good representation of, because indeed they start with developing the high blood pressure, obesity, and diabetes, which is a major cause for the other pathologies.
Mm-hmm. But if you're then going to find, so you found specific genes, right, that are differentially expressed. So do you think they are contributing to one specific effect of a comorbidity, or are they a result of, indeed, the combination of those comorbidities?
I think it is a combination of the comorbidities, because there is a general high stress on the vasculature and endothelial cells, and
Mm-hmm. So when targeting those genes, because I guess that would be a next step in the study, therapeutically, I mean, do you think, again, you will target or you will enhance cognitive function, or will you also be targeting the comorbidities themselves?
I think both. I think TRPV4 and KLF2 are important in generally the health of the microvasculature. So by targeting those, you will improve the function of the vessels, but it can also indeed affect the other organs. I think a study on Alzheimer's disease in mice, they studied the effects of PIP2 administration in this model, and they showed, which is a regulator of TRPV4 actually, and here they show an improvement in the cognitive function. So I think by regulating these, it can have a generally beneficial effect.
Okay. Thank you. My next question is on one of the major findings in Chapter 2, which is that you found a higher vascular density in the obese rats, but also higher size of leakage areas. So I think you already mentioned that there might be some compensatory mechanism there, but do you think there is also just a correlation between higher vascular density and more leakage?
Yes, this is possible. I think when the BBB leakages start developing, the neurovascular unit is dysfunctional. So the neurons, they do not receive the needs that they are asking for. And I think these trigger the angiogenic mechanism that leads to an increase in vessel density.
Maybe. Or the other way around? So could leakage be a consequence of angiogenesis?
That I don't know.
Okay, something to think about. I still have some time. Some other questions about, again, vessel density. And you mentioned in Chapter 4 that it might be important to explore differences in vessel density in different areas of the brain when you were mentioning your 3D imaging techniques. So that made me wonder, does vessel density correlate to neuronal activities then in different brain areas?
I think it's a relevant question indeed. We have not checked this, but I think it would be really interesting to look into the different areas of the cortex and see if it aligns with differences in vascular density. Yeah.
So, and maybe then translating to the disease that you're modeling. So do you know in CSVD whether this happens in specific brain areas, whether the pathology is localized to some preferential areas in the brain?
I think in CSVD it is mostly happening in the white matter as well. So they see a lot of damage in the white matter and lesions, but also small microbleeds. But they also see atrophy from the cortex. So I think a lot of different brain areas are being affected in this disease.
Okay, thank you. I give the word back to the prorector.
Thank you very much for your opposition, Dr. Broeks. Then we come to Professor Lutten. He's Professor of Medicine in the Department of Cardiovascular Science Center for Molecular and Vascular Biology in the University of Leuven. And thank you for coming all the way from Leuven.
So first, also like to start off by congratulating you with a very nice thesis manuscript, and I would like to extend those congratulations to the promoter team. Also, your presentation was very didactic, so congratulations on that. So I would like to start off with a very introductory question. So one of the most important findings was that you had neurovascular uncoupling in your model. So one of the important mediators of coupling is NO. So could you elaborate on the different sources of NO that are involved in that, and whether there would be difference in their relative importance in coupling the neuro to the vascular?
Highly esteemed opponent, thank you for your compliments and for your questions. I think, yeah, indeed, NO can be produced by different cell types in the brain, by the neurons, vascular smooth muscle cells, endothelial cells, and it has been shown indeed that I think endothelial cells are most important in actually vasodilation. So I think in there it would be most important.
Okay. So you use the rat model because it's a very exceptional model and one that has nice complement of comorbidities. So suppose you have an interesting gene that you would like to study in cerebral vascular disease, and you have a knockout model for it. So would there be an equivalent way of reproducing all these comorbidities in a mouse and then study that, or is that a difficult situation?
That was initially the plan after identifying the genes TRPV4 and KLF2. I think a knockout model is indeed interesting to study very specific effects of only this molecule, but it's not completely representative for the full model of comorbidities. So I think clinically it is more interesting to look at a model of comorbidities.
Mm-hmm. Okay. So TRPV4 is one of the important targets that you found as a downstream mediator, possibly of the vascular dysfunction. So suppose you would like to design a therapy based on this target. So knowing that it is there in several cell types of the neurovascular unit, and so how would you go about that to design a therapy based on TRPV4 to solve the problem of?
I think this gene first has to be studied further in vitro to see the action of agonists and antagonists on the vessel, on the endothelial function, on the barrier function. And I think if you would have to translate it clinically, there have been some antagonists already being tested in heart failure patients, so it has been shown safe. It hasn't been studied yet in neurological disorders, but currently they are testing it, a TRPV4 agonist, on the neurovascular coupling in dermal vessels, so it would be very interesting as well. I think if you really want to specify TRPV4 as a therapy, I think it would be interesting to look at PIP2 that regulates the working of TRPV4.
Mm-hmm. Okay. So another question is, one of the observations was that the ejection fraction in your obese ZF1 rats was actually increased. So it is a model of non-reduced ejection fraction, but it was actually increased. So what would be your hypothesis about the reason behind this?
Yes, indeed. We would still expect the ejection fraction to be above 50, but not to be higher than in the lean animals. We think that the heart is actually trying to compensate for the poor relaxation so that it is pumping harder to still reach the ejection fraction or the blood that is needed to pump around.
Mm-hmm. So then maybe my final question, if I may. So there is, so the microvascular dysfunction is a common underlying phenomenon in the heart as well as in the brain. So your idea is then to try to find a way to remedy that in both organs at the same time. But how would you take into account the fact that these vessels are very different in these different organs? Would that be a complication of trying to fix the problem in these two very different organs in terms of their vessels?
I think microvascular dysfunction is something general, so it would be interesting to look at TRPV4 as a general improver of the health of the microvessels. I think the baseline properties of the microvessels are still similar in most organs, so I don't think it would be a problem.
Okay, thank you. And now I give the word back to the President.
Okay, there's still time, so first of all I go back to the Secretary, Professor van den Hove. You have additional questions in the opposition?
All right, thank you. That was rather unexpected, never happened, but it's absolutely fine. So may I ask one of the parliaments to read out statement number 8? Normally we're always short of time. That's why I was surprised. Yep.
The part can never be well unless the whole is well.
So there, when I read this one, I was also thinking of Tolstoy, who says something that is maybe opposite to this, but it can be merged. So that statement relates to all happy families being alike and all unhappy families being unhappy in their own way. Meaning that if you translate that to biology, anything that could go wrong could trigger somebody to become unhappy, or in this case, to show cognitive deficits or any related phenotype. So can you maybe comment on your own statement and possibly see where this links or matches with the one I just presented to you?
Mm-hmm. I think with this statement, I mean that indeed we are looking at CSVD and HFpEF, but the microvascular function is probably affected in a lot of other organs too. I think by just finding a treatment to improve cognitive function or heart function, you will never solve completely the problem. So therefore, I think we should be focusing on general restoring the microvascular health in multiple organs and only focusing on one disease.
Yeah, so that links a bit to this discussion that you had earlier down the line,
yes.
saying that the players could be involved in many of these processes.
Yes.
Perfect timing.
Thank you, Professor van den Hoven, for your opposition. Dear candidate, Ms. Lambrichts, the time appointed for the defense of your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and the way you defended it. And I request that you and your company here on site and online await the results of our deliberation and our return in this room. With it, I suspend the meeting.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Can't fly by now, don't waste all your time. Cause I'll go the extra mile.
I reopen this academic ceremony. Ms. Lambrichts, dear candidate, the degree committee here present has assessed the quality of your thesis and the way you defended it. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And your supervisor, Professor van Oosterbrugge, is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And I invite Professor Oosterbrugge to now take the floor.
Dear candidate, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
Great. By the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, Sara Maria Paula Lambrichts, the degree of doctor and grant you all rights attached by custom and law. As evidence to this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Dear Dr. Lambrichts, dear Sara, it's a privilege for me to congratulate you as the first one. You did two journeys, a personal one and a scientific one, and both brought you to the top of the hill. It wasn't always easy, sometimes it was a bit difficult, but you always managed to get your way back and find at last, and with big success, the way up on the hill and found the top. And although I was a bit bystander as a clinician with this, for me, difficult work in the laboratory, I think fundamental for this journey was the support of Sebastian, and it's my pleasure to give him the mic, to give him the honor to do the laudatio.
Thank you, Robert. Dear Dr. Sara Lambrichts, dear Sara, I'm very proud to stand here and to congratulate you together with Robert, and to also reflect on your academic journey. You began your PhD in March 2020, a time we all remember as the world entered the lockdown. It was an uncertain, disorienting period, and yet you entered the research world with a strong commitment. From the very beginning, you were the integral part of the European Crucial Project, a highly interdisciplinary collaboration involving partners across Europe. You engage with teams from Professor Liz Jones from KU11, Professor Mieke Deverschen at the VIB, along with colleagues from also UK and Spain. And all these collaborations expose you to a wide range of expertise, and you listen and discuss with neurologists, including your own promoter, radiologist, molecular biologist, physiologist, pharmacologist, imaging experts, and this very broad expertise also is nicely represented here today by your jury members. And you embraced this diversity with curiosity and openness. But the project was not easy. It was demanding, at times overwhelming, requiring you to move across disciplines, combine techniques, and dive into unfamiliar territories. I know there were moments where you doubted yourself, where you left the weight of the work ahead, moments where things were too vast, too complex, out of reach. But you kept going, as also Robert just said. You found your own pace, and yet you moved forward, step by step, finding your way through. You learned to focus on the smaller steps, the ones that slowly but surely brought you to where you stand today. Along the way, you use techniques from behavior neuroscience, from cardiology, from microscopy, biology, vascular biology, molecular analysis, and no single person can be an expert on all of these fields. That's also what we discussed together. But you brought them together meaningfully with scientific rigor, and you showed how interdisciplinary work is important in this field of research. So working as a team, navigating, integrating, making choices. That's the academic life. Of course, some challenges were more personal than technical. I still remember you were telling early in your PhD, quite honestly, I don't like working with computers. And I was then very worried, I have to say. And yet, by the end of the project, you had learned to perform advanced image analysis and even took part on the bioinformatic data processing with the support of colleagues from toxicogenomics and FSE. You didn't love it, but you did it, and I hope you're proud of that as well. There are many more achievements worth to mention, and I'd like in particular to highlight that you have presented your work at many general assemblies and conferences, like at the FENS in Paris, several editions of the Dutch Neuroscience Meeting, and also during the Euron PhD Days, where you actually were awarded the first prize for your outstanding presentation in 2022. These are not just accomplishments, but also moments that show how much you've grown into your own space as a scientist. There's also something I would like to say, something I also written down on a card you will find later, and I'd like to refer to it here in a more philosophical way, because after all, that's PhD, that's a doctor for philosophy. The PhD is not simply about gaining knowledge, but as you've discovered, it's about transformation. It's about learning to draw your own lines, and while you were shaping your own journey, you also were shaping ours. The ones of your colleagues, your friends, and mine too. So I've grown too as a mentor and a person while also witnessing your own growth. So thank you for that. And on a slightly lighter tone, I also would like to highlight that the way you shape this journey goes far beyond your work at the bench. Anyone in the building could tell when you were around. Your laughter echoed through the corridors and filled the lab with life. Your dedication to your fluffy friends, the rats that you work with so carefully, they also showed your compassion and the sense of your responsibility. You always went the extra mile to ensure their welfare, and that quiet care also defined the way you worked with us. I was not surprised to see that the rats appear on the back cover of your book, and also here on the screen, on every new chapter page. But beyond these fluffy friends, you also form meaningful bonds with human beings, of course, with many members of the team, Eline, Narek, Melissa, also people beyond our department, internal medicine, all the interns, and especially Irene, watching us from Madrid, and obviously your close friend Laura, both scientifically and personally. Laura was also a very important partner for this ZSF1 project, and much more beyond that. And later also, when Hélène joined the department, she too became a key companion in this journey. So together they stand with you as your parent here, a reflection of your friendship, trust, and deep mutual respect. And while we are talking about your presence in the lab, still, we cannot ignore the legendary corner of your office. Yes, your desk, or should I say the ecosystem of papers, tubes, mysterious items, Kinder Chocobon, I noticed last week. Who knows what else that seemed to grow and evolve by the day. I have to confess that every time I stepped into the office, I expected to suffer from a heart attack. It was truly a hazardous zone for anyone with a need for order, and a true test of patience for your office mate. I'm thinking especially of Narek, who, let's just say, had very strong feelings about that. But somehow, still, with all the chaos around, you always knew exactly where everything was. At least that's what you claimed. But Sara, it's now time to let you go. You are ready for the next challenge, your next transformation, I hope. So thank you for everything you've brought to the team, your energy, your laughter, and your kindness. And as a final note, I'd like to refer to Anthony van Leeuwenhoek, a famous Dutch scientist often referred to as the father of microscopy, he showed us that true transformation begins when we learn to see differently. As he once wrote, sorry for the Dutch, hoeveel is er niet te ontdekken? How much there is still to discover. And I believe, or at least I like to believe, that this doesn't only apply to science. I think there is still so much to discover about ourselves and about yourself, so I hope you can have this opportunity in your next step. With that, van harte gefeliciteerd. Congratulations, Sara, and well done.
Thank you, Dr. Foulquier, for a very nice laudation. Dear Dr. Lambrichts, also on behalf of the University of Maastricht, I would like to congratulate you with the degree you have acquired, and of course, congratulations for your parnames as well, and for your supervisors, Professor van Oosterbrugge, Dr. Foulquier, and of course, family, friends, colleagues. All congratulations, and I hope you can celebrate it in the right way, because probably this is one of the most important days in your scientific career, in any case. Having said that, we are going to close this meeting. I would like to thank the opposition, of course, the members of the opposition for their work, and the PEDEL and the technical support team who made this ceremony possible. I have one last announcement. I can probably do that in Dutch. Dames en heren, want de meeste van u zijn Nederlanders, geloof ik. Althans, zijn Vlamingen ook Nederlanders. Verstaan Nederlands, laat ik het zo zeggen. Wij gaan zo de zaal verlaten in cortège met de paranimfen, het certificaat en de jonge dokter. Dan gaan wij een foto maken aan de voet van de grote trap die u hebt gezien. Ik zou u willen vragen niet allemaal te gaan kijken, maar vast naar de refter te gaan, waar de receptie is. Dan kunt u alvast een drankje nemen of een kop koffie, wat u wilt. En dan komen wij daarna wel op de foto met u en eventueel een tweede foto op diezelfde plek met de familie, maar die moet dan even meegaan. En de fotograaf natuurlijk. En als u dan denkt, wanneer kunnen wij de kandidaat feliciteren, de jonge dokter, dan is het heel simpel. Wij komen op een goed moment ook naar de refter toe en dan gaan wij eerst en dan komt u. Dus u houdt ons gewoon in de gaten. Dat gezegd hebben, sluit ik deze ceremonie.